So today we'll go with very interesting and amazing topic, which is about packaging of DNA. So let's check it out. What would be before that? You should always know about DNA. I have covered so many videos, but still, DNA, which is deoxyribonucleic acid, which is simply acidic, right? Acidic. This means their pH would be uh, 7 is neutral, from 1 to 7 and till 14. So from 1 to 7 it would be acidic, and from 7 to 14 it will be basic or alkaline. So basically DNA is deoxyribonucleic acid double helix structure which have nitrogenous bases. Nitrogenous bases are what? Adenine, guanine, cytosine, and the last thymine. These four nitrogenous bases are present, right? And even the DNA also have, like, when we talk about the composition of DNA have three composition, like made up of three things: nitrogenous bases, second when we talk about called as pentose sugar, and the last one called as a phosphate group. These three things. So our today's topic is about packaging of DNA. So of course you would be none, our DNA is quite long. I mean, this have literally so many things. If we talk about the particular length, DNA is around 3.4 meter long or 34 angstrom. And when we talk about the DNA length, we write in a dot. Even the DNA length, when we talk about that's also called the pitch of a DNA. So here we are going to start basically our today's topic. So if it you might get this question in your mind, that when we talk about if DNA is literally 3.4 angstrom long, so how is that remaining in our chromosome, right? So let me just explain you the packaging now. DNA, which we know that's acidic, DNA acidic molecule, acid molecule. So there we need something to control it out. So if this the DNA, this is going to get fold, but of course acidic can also not survive. So what we have something extra. Then we have a protein like what? We have protein which is called as a histone protein. Why we have only chosen histone? Because that's a basic protein. To control acidic environment we need basic protein. And when in the histone, like in this one, we have two basic protein called as lysine protein and adenine protein. These both protein are basic protein which are used for DNA to control it out. So before that, even histone, when we talk about the histone detail, histone protein, which is simply making a structure which is called as octamer. This is an important MCQ. Octa word tells it's related to 8. So yes, this is 8 like a structure, form like a structure like this one, something like this one. This means our 8 structure will be formed, right? So I hope it will be clear now. Histone protein, which is again divided into few more part, few other proteins, not proteins but basically histone type. So when we talk about the H1, second one called as the H2A, third called as the H2B, and the fourth one called as the H4, last one called as H5. These 5 histone proteins are basically present. So what is the function of all? I'll just let you know that as well. So these 4 are all together double like structure like this one, and are used together except for H1, which H1 function we'll do later on. So here comes our structure which we are going to discuss. This the structure, right? So here you'll get the term. Here, if this the DNA, of course, if this the structure, and here is the attachment of protein. Which protein? Of course histone protein. That's DNA. DNA plus histone. So whenever you just see DNA plus histone, then we give them as a term that's basically called as a nucleosome. So never get confused about the nucleosome. Nucleosome is simply when DNA plus histone are combined. Why we are doing that? Because DNA is quite long. DNA is 34 angstrom long, right? So we need something so that it can be bounded around. So when they get histone protein, they get bounded, and histone protein is basic. That's why they literally get tightly bound. Then the same process goes here. The histone protein get attached with so many things, right? Like this is structure. So nucleosome is found, right? Then comes another part. Here the one part where they're not attachment of stone, like for suppose here's the no attachment of stone, what it will be called? So whenever you just see no attachment of stone, at that time there's that's called as a linker DNA. Linker DNA is made up of H1. Now here H1 protein, H1 stone is used. So for linker, remember always H1 stone would be used, not any other. Rest for entire H2A, H2B, H4, H5 would be used. I hope it would be clear then. Then here we have this kind of a structure. This structure is simply called as chromatin. Chromatin is also called as a something else that's called as a beads on a string, right? So I hope it would be very much clear all these things. Then comes the next, which is about what here around how many nucleosomes are present on the base pair. Like oh wait, the question is how many nucleosome are present on the base pair. So from 1 to 200 nucleosome would be present per base pair. From 1 to 200 nucleosome would be present on the base pair. I hope it will be clear. Then comes another part which is about nucleoplasmin. So if this is the structure, if this is the DNA, here's the structure, here the part where and the structure is here, and DNA would get fold like that. So the one at this DNA get folded, that's called as a nucleoplasmin. So even then this is going to get attached in that chromosome. In the center we have other parts. Here what will happen? In this one middle there will be non-histone chromosome protein present. This is non-histone chromosome which is present. Then what we'll see, there would be this part is going to attach throughout end of this one, and then yes, here comes the entire division or packaging, everything would be done. So this is called as a packaging of DNA. What we discussed, DNA acidic condition, we need something which is made up of basic. We got histone protein which is lysine and adenine. These both histone is of different types, made up of octamer, 8 other structure, packaging, nucleosome, everything. So yes, here comes the end of this video. Thank you for watching. Bye bye.